Now, yes, but for the moment we need to leave them down only for the questions to open them.
Yes, but I need to do the introduction.
Okay, you want to start?
I don't know.
See, now it's 15, so I think...
It's almost time.
I think so.
No, but it's already broadcasting. Maybe we could start. We are connected. Yes. Since half an hour already. More, there's maybe 1 hour. No told me that it was working. That it's online. Check. Yes, I did. That it was online.
So then, when we checked upstairs, it was already like saying in 19 minutes.
Yes.
It's okay. Perfect.
It's online. Do you want to introduce Clive?
No, I won't tell.
Do you want my text to
Oh,
introduce
have you
you? No, but you know him. No, please, please, do it.
Go ahead.
Do it.
Go ahead. You can talk whatever lies you wish.
But,
So Louise can introduce you.
So, hi everybody. Can you all hear me? Okay. So, today we are welcoming Dr. Clive Truman. He's going to talk to us about stable isotopes in fish and in fisheries research. Dr. Clive Truman is Associate Professor at the University of Southampton. He did his bachelor as well as his PhD in Bristol, and is specialized in geochemical ecology and biominerals chemistry. As a marine ecologist, Dr. Truman uses stable isotope approaches to explore aspects of trophic, spatial, and metabolic ecology. Dr. Truman and his research group in Southampton has been leading the development of marine isoscapes and using this to track the movement of individual animals across isotopic gradients. The ecological subjects of Dr. Truman's recent research include bony fishes, sharks, turtles, seabirds, and marine mammals, mostly focused in European and Northeast Atlantic waters. He has over 100 publications in peer-reviewed literature. Thank you, Dr. Truman, for coming here to the Stazione Zoologica, and the floor is yours.
Thank you very much. Do I need to talk into this one? Okay, thank you very much. Thanks for inviting me, and thanks for coming, and also to people I can't see. Okay, so hopefully this will all work. So what I want to talk about is the application of stable isotopes in marine ecology, not just in fish and fisheries, but in marine ecology in general. So when we're talking about stable isotopes, at the really fundamental level, we're just talking about something beautifully simple. So here's carbon, an atom of carbon with 6 protons and 6 protons and 6 neutrons, carbon 12. An atom of carbon with 6 protons and 7 neutrons, carbon 13. And everything we're talking about is just this little neutron. This real simplicity that we have one extra, in the case of carbon, one extra neutron, which is giving that atom more mass. It's making the reactions that that atom is involved in behave with slightly different kinetics, different bond strengths. And the presence of that extra that extra neutron gives rise to a whole range of pattern in natural systems that signal that we can exploit. And I think this is just such a simple thing that leads to so many different scientific potential, so much scientific possibility. And that's really what makes me excited about using stable isotopes. Starts very simple, you can do a lot with it. So, let's think about what we can do. So, the majority of work in isotope ecology is really associated with using stable isotopes to say something about diet or something about trophic ecology. And fundamentally this works because with that extra neutron, these molecules with the heavy isotope have higher bond strengths and they behave with different kinetics in reactions. Generally, we synthesize excretion products with more of the light isotope, and that means that as we go up a food chain, we progressively increase the proportion of heavy isotopes in our tissues. So this leads to a very sort of fairly familiar pattern where the isotopic composition of consumer tissues increases in these delta values that we express isotopes, increases as we go up the trophic level. And there is a huge literature exploring the use of isotopes to say something about trophic ecology, about trophic level, food webs, diet resources and diet sources. And I'm not going to talk about that today, because I want to talk about other applications of stable isotopes that might be less familiar. And I want to focus in on what's happening at the base here. Because if we have a single resource, then we have this nice simple relationship between the isotopes of carbon and nitrogen and trophic level. But as soon as we allow the primary production to change its isotopic composition in time and space, then we have complexity for understanding the trophic applications, but we also have signal that we can explore in a spatial and temporal context. So what I'm going to talk about today, I'm going to have sort of three main applications that I want to sort of think about. We're going to think about applications of isotopes in ecology, marine ecology, and I'm particularly going to talk about using isotopes to explore migration, to think about connectivity, and to think about the role of animals in nutrient flux. I'm going to think about isotopes in fisheries, largely through the idea of traceability and food forensics. And I'm going to talk about the use of isotopes in predicting how species might respond to global warming or warming of waters. Okay. So, as we said, first of all, we've got variation at the base of the food chain in the isotopic composition of primary producers. And in a marine context, we have variation because we have different packets of water, different current systems. And we also have different temperatures. And here you're looking at the amount of primary production. And we can see that we've got changes spatially, big gradients in the extent of primary production. And we also have variation temporally. And those spatial and temporal variations in the extent and distribution type of primary production are going to give us isotopic signal. It's isotopic signal that allows us to say something about spatial ecology, about movement, because the isotopic composition of something here is going to be different to the isotopic composition of something here, because the isotopic composition of the primary producers varies. But that's a basis behind using isotopes in spatial ecology. Okay, so the first set of applications that I want to explore is quantifying animal-based nutrient flux. So this is the idea that animals are responsible for fluxing and moving nutrients between ecosystems or across ecosystem spatial boundaries. And we might want to be able to quantify that. So the first example is actually a really beautiful figure from a really nice, quite early paper exploring how isotopes can help not just in quantifying nutrient flux, but actually explaining pattern. So I just want to run through this picture because it's also a really beautiful graphic. So this is a study looking at the consequences of changing tropical vegetation from native vegetation to oil palm vegetation. So what you can see in these bar charts is basically a contrast in the amount of nitrogen under native and palm oil plantations. So you can see that native trees have more, that's actually much more percent cover than palm trees. Native trees have more biomass than the palm trees. And the soil nitrogen is higher under native tree conditions than palm trees. And also the leaves have more nitrogen, there's more nitrogen in the runoff, there's more nitrogen in, and there's more zooplankton, and there's more manta rays, where you have native wildlife, native trees compared to palm oil plantations. And when you also look at the nitrogen isotopes in soil, leaves, waters, in this case sponges and zooplankton, what you're looking at here is the difference in isotopic composition in soil nitrogen between native vegetation and palm vegetation. And there's a very big increase in the delta 15N values in the native vegetation compared to the palm oil plantations. And that happens because marine nitrogen, the marine food chains are longer, these are size-based food chains, and so the delta 15N values in marine ecosystems, you can get very high delta 15N values. And effectively what this whole study is showing is that where you have native trees, you have seabirds nesting in those native trees, excreting marine nitrogen, and that marine nitrogen is then flowing through the system. When you remove the native vegetation, you have palm trees that birds don't roost in, and so that ecosystem flux of marine nitrogen into the terrestrial food webs is disrupted, and one of the downstream consequences of that is that you have less zooplankton and less manta rays. There's a really nice explanation or demonstration that one perturbation, one change in an ecosystem can result in an unexpected consequence because of the animal-based vectoring of nutrients from one system to the next. And the isotope part of that it's a really nice demonstration. Let me show you another example of that. So there was a horizontal nutrient flux. So I want to show you something that we worked on a while ago as a vertical nutrient flux in deep sea systems. So we're going to move now to the west coast of the UK, the British Isles, and we're looking at the continental slope here. So there's used to be a large fishery, a deep water fishery on the continental slope of the UK. So we're now looking at fish assemblages from 500 meters water depth down to 2,000 actually. Here I'm only going to talk up to 1,500 meters water depth. So relatively deep water fisheries. And what I'm showing here is the isotopic composition of demersal fish, so fish caught within 20 meters of the seafloor at 500 meters, 1,000 meters, and 1,500 meters water depth. So quite deep. In red, you have the isotopic composition of benthopelagic fishes, so that's fishes that are feeding away from the seafloor, but still caught close to the seafloor. And in black, benthic feeding fishes, so fishes that are actually strongly focused on foraging at the seafloor. The blue star is approximately the isotopic composition of zooplankton in this region. So hopefully what you can see is that, as you'd expect, as the water depth increases and the physical separation from primary production increases, so the isotopic composition of the benthic feeders is getting further and further away from primary production. And that's basically telling you that there are more trophic steps involved in transferring nitrogen from primary production to fishes at 1,500 meters than there are at 500 meters. That's what you'd expect. But for the benthopelagic feeding fishes, actually, there's no difference. The benthopelagic feeding fishes have the same access to resources at 1,500 meters than they did at 500 meters. And at 500 meters, the benthic feeding fishes have the same access as the pelagic feeding fishes. And the way that we can explain that is that we have a large-scale vertical migration of things like myctophid fishes and other parts of the mesopelagic community that move between 500 meters water depth approximately during the day and up to the surface at night. So this community is providing a nutrient flux that makes surface production very immediately accessible to bottom water or bottom-dwelling fishes and bottom-foraging fishes at around about 500 meters. By the time you get deeper than the daytime depth of this community, the benthic feeding fishes no longer have direct access. And so the isotopic composition starts to increase because they're needing an extra trophic step to get from here down to here. But the benthopelagic feeding fishes are still able to move up and access this community and flux it back down. Why does this matter? Well, this matters because water depths below about here act as carbon sequestration. So carbon deeper than around about 500 meters water depth is no longer able to, or no longer efficiently able to flux back to the atmosphere. So anything that's capturing carbon above 500 meters and moving it down to sort of 1,000 meters or below is providing a carbon capture and storage ecosystem service. So if we can identify that service provided by, in this case, the benthopelagic feeding fishes that live below the sequestration depth, move up and intercept the animals that move up to the surface, capture their carbon and take it back down, then we can quantify the carbon capture and storage service provided by this part of the community. We can capture that and we can calculate its value in terms of the carbon trading value. And we can measure it spatially if we know what the biomass of those fishes are. So we estimated the carbon capture, the extent of carbon capture provided by benthopelagic vertically migrating fishes in this region. And this is a fairly substantial carbon capture service. We can turn that into a financial value, and we can compare the carbon capture and service value to the fishery value for the value of the landed fish. And what we find effectively is that the carbon capture ecosystem service value is greater than the landed value of the fishery. And that's the landed value, not the profit. This is a very heavily subsidized fishery in terms of fuel subsidy. And when you take the subsidy away, then the ecosystem service value is hugely outweighs the fishery value. So this was part of information that went into EU lobbying to get the deep water trawling limitation put in place, which was successful a few years ago. Okay. So I want to move to a second application of isotopes now, and this is thinking about isoscapes as a basis for spatial ecology, which is actually most of what I'm going to talk about. So what we looked at just now was an application of movement of nutrients from one isotopic composition to another, one area with one isotopic composition to another with a different. What we want to do, what we're interested in now is expanding from just one or two places to having continuous measurements of isotopic variation throughout space. So work that we've been doing with initially a PhD student, now postdoc in Southampton, Katie St. John Glue. That's what I'm going to use as a demonstration of marine isoscapes and what we can do with marine isoscapes. So if you want to capture the spatial variation in stable isotopes across a marine region, you have to choose some kind of organism or material that you're going to use to measure the isotopic composition in known regions. And in open marine systems, especially in pelagic systems, it can be hard to choose what your reference organism or your reference material is going to be. So we've decided that jellyfish, or the Scyphomedusan jellyfish, provide a really useful target. And one of the reasons they provide a really interesting target, a helpful target, is because they're essentially, in the most part, growing their tissues in one season, and they have a diet source that is similar to most of the organisms that we're likely to be referencing to our isoscape. In other words, they're feeding on zooplankton, fish larvae, and to some extent larger fish. So a similar diet composition to most of the animals we're thinking about. They're also very evenly spread across our waters and relatively easy to sample. So it makes a nice target for building an isoscape. one of the downsides of building a large spatial isoscape, and here we're over 1 million square kilometers that we're trying to map. And one of the problems that you have when you're dealing with a relatively large system like that is that there's rarely one species that covers the entire area. So we have to have some way of incorporating the isotopic variance that's associated with different species and accounting for that, as well as the spatial variance that we're interested in. Katie's been using a method called INLA. I'm going to have to remember this. Inverse nested Laplace approximation, which is effectively a Bayesian version of mixed models, to produce effectively a model of the spatial variance, or the spatial component of isotopic variance across all of this region that factors out the species effect. So she sampled around about 700 or 800 individual jellies of 6 different species, and here you can see the species composition in broad regions of British Isles waters. So without going into a huge amount of statistical detail, this is effectively what you get as a isoscape model, and I'm not sure the colors have rendered particularly well, but what hopefully you should be able to see is that we have a continuous surface that predicts or explains how the isotopic composition of carbon and nitrogen, we did also measure sulfur, varies across this seascape region. And I should point out that this range, 5 per mil in carbon and what's that? There's actually nearly 8 per mil in nitrogen. That's a big, big range. So this is a large spatial signal that gives us signal to explore aspects of movement and migration. It also provides complication if you're interested in using isotopes for diet analysis, because you would need to take into account the range or gradients in stable isotopes if you're trying to compare trophic level from here or here. Or for instance, if you're interested in the variation in isotopes in a particular community of animals that straddles an isotope gradient, you would need to take that into account. So these isoscapes, these isoscape models give us insight into spatial distribution. They also give us insight into the mechanisms that underpin that spatial distribution. But one other thing that we can do, if we've taken out the species effect to explore the spatial effect, which is what we do with our INLA modeling, then we can also explore what that species effect is. So here, what these distributions represent is effectively, it's the effect of species on the isotopic composition when you take out the spatial component. And you can use this to explore or to ask, why do you have a particular species composition in the places that you do? And we can see, for instance, if you look at nitrogen, you can see this species, which is the mauve stinger, and this species, the crystal jelly, so here and here, are really widely separated in nitrogen isotope values. You can also see that we see these two species, they actually occur together. So we're seeing trophic separation, allowing these species to occur together. Other species, much more similar in their isotopic composition, suggesting that there's more likely to be competitive exclusion, and particularly between the lion's mane blue jellies. We see that. We see that we don't usually see lion's mane blue and compass jellies present at the same time. So we can use the spatial component that we are interested in as our signal for an isoscape. If we extract that, then we can look at the ecological signal as well. Okay. Well, what can we do with an isoscape once we've got it? Well, one of the nice things that we can do with an isoscape is we can assign an animal to the area that it was probably foraging in, or likely to be foraging in. And the way, the sort of flow chart that we might go about to do this, we have an isoscape which we can break down into a grid with a predicted isotope value in each square of the grid. We have an isotope value that we measure from the animal that we want to assign to its likely location. We measure the difference between the measured value in the animal and each cell of the isoscape. And we can then relate that to a probability density function if we know the variance of the isotope values across the whole surface. And we can extract the cells that have a certain threshold level of likelihood based on that probability density function. We can then map out where those cells with high similarity, high probability are, and produce a likelihood map of the area that that animal was foraging in. I can give you an example now with seabirds. This is another of Katie St. John Glue's papers. So this was looking at guillemots, razorbills, and puffin from the Isle of May, which is in the Firth of Forth, around about here. So here we have feathers from birds that have also been instrumented with light-based geolocators. So people are interested in knowing where these birds have been foraging in, particularly in the winter when they are not present on the Isle of May. So, light-based geolocators can give you some indication of where an animal has been foraging with a degree of error. An isotope geoassignment will give you an idea of where the animal has been foraging with a degree of error. But those are two completely independent methods. So we can combine those two methods and use one as a prior, or a Bayesian prior for the other, and reduce the uncertainty to get a more precise estimate of where each animal has been foraging. So here in green is the kernel density area of likely foraging from the light-based geolocators, and purple is where the isotopes suggest is the most likely foraging region, and so we can highlight the, if you like, the highest likelihood area, combining information from both of those independent proxies. And one of the really nice things we've been able to do with that approach is explore the responses, the foraging responses of two birds from the same location under different conditions of environmental stress. So here we have puffin and razorbill. And this is a time series of survivorhood based on basically reciting known pairs of birds over a long time period. So you can see that in 2014, we had a kind of average level of return rates, an average level of mortality. It was a relatively good year for both puffin and razorbill. In 2007, it was a really high mortality year, really low survivorship for both for razorbill, but especially for puffin. So there's a question of why. Why do we see this really high mortality event in 2007? And is there something different in the way that these animals were foraging? So this now is the combined likely area of foraging based on the isotope and geolocator information. And so this is the likely areas of foraging in the good survival year and the likely areas of foraging in the poor survival year. And so what you can see is that in a good survival year, puffin and razorbill that are coming to the Isle of May here, both forage close to where their nesting sites are. Okay? So they're foraging in productive grounds and they're not having to travel very far to forage there. In the year of poor survival, the puffins stayed foraging close to where their nests are. So they didn't change their foraging distribution. The razorbills really changed where they were foraging. They moved much further south. And I'm not sure if I put the next slide back on here. No, I didn't. So what actually happened in that year is that there was an outbreak of snake pipefish, which is a small pelagic fish, which essentially replaced the normal diet of sand eels in this location. And effectively, the puffins stayed where they were and foraged on low-quality food, whereas the razorbills invested more effort in migration to be able to forage on high-quality food. And in this year, that difference in the strategy response to resource limitation was a better strategy for razorbill than the staying in the same place and foraging on low-quality food was for puffin. There's a really nice bit of information that we're able to extract using retrospectively from the isotopes of the feathers of these birds. So this is the likely area of foraging based on the isotope values across the whole surface. And what you can see is that in a good survival year, puffin and razorbill both forage close to where their foraging responses are. So here we have puffin and razorbill. And so what you can see is that in a good survival year, both forage close to where their nesting sites are. Okay? So there's a really nice bit of information that we're able to extract using retrospectively from the isotopes of the feathers of these birds. Okay, so I go back to fishes, and the next thing I want to think about is what information can you get from incrementally grown tissues? So can we say something about the life history of an animal from a tissue that's grown throughout its life? So now we're going to look at bluefin tuna. I had to think of bluefin tuna because I'm close to places where bluefin tuna are. So this is a nice example. So this is an eye lens. This is the eye lens. It's not of a bluefin tuna, actually. This is an eye lens of a deep water fish. But eye lenses are crystalline proteins, and they grow incrementally. And you can't afford to have cellular tissue in your eye lens, otherwise it's going to diffract light and it won't work. So eye lenses are acellular, which means that they can't be regenerated or regrown after deposition. So an eye lens core is the tissue that was laid down, in this case, as an egg or an early larvae, and that continues to increase in mass as the animal grows. It's made of protein, so we can peel that eye lens, and we have an isotopic record through the life of the individual fish. And we can explore what a fish has been doing throughout its life based on the isotopic composition of its eye lens tissues. So we're going to have a look at Atlantic bluefin tuna. So we have the supposed spatial distribution and lifestyle of Atlantic bluefin tuna, summarized crudely in this diagram. There's two main spawning areas, one in the Mediterranean, one in the Gulf of Mexico. And Atlantic bluefin tuna conduct large-scale cross-Atlantic movements and migrations. And this makes them difficult to manage in a stock perspective. So we really want to know quite a lot about what proportion of individuals move from, let's say, a Mediterranean juvenile origin. How much time do they spend foraging in the east coast of the US compared to the eastern, northeast Atlantic before returning back to spawn? A lot of work has gone on in putting satellite tags on bluefin tuna, but it's hard to put a satellite tag on a larval fish. Well, you can't put a satellite tag on a larval fish. And it's hard to, and satellite tags don't usually stay on for 5 or 6 years. Never stay on for 5 or 6 years. So we don't get very good, although we get obviously very high precision data from satellite tags, we don't get long-term data and we don't get juvenile data. So we had a look at eye lenses of bluefin tuna caught in bycatch fisheries in the English Channel. But to understand what they're telling us, we have to have some isotopic context because we don't have an ice escape for the Mediterranean and the North Atlantic. So the best we can do to understand, to get some isotopic context, is to look at large data sets of the isotopic composition of tuna caught across the North Atlantic region. So here is a data set of isotopic compositions of tuna from the eastern side of the North Atlantic, so Bay of Biscay up through to Ireland. Here from the western North Atlantic, so from the eastern seaboard of the US. Here from the Mediterranean, largely from the Adriatic, but also close to the Balearics. And here from the Bay of Biscay. So we have a nice range of isotope values, but specifically longitude is a very clear difference. So the US side have relatively positive delta 13C values compared to the eastern North Atlantic. So that gives us some signal to compare. So now what you're looking at are the eye lens values of 30 bluefin tuna caught in the English Channel. So the point size is relative to the reconstructed fork length of the fish, which we can reconstruct from the eye lens. So juveniles down here, and this is consistent with what you would expect a juvenile or a larval bluefin tuna to look like in the Mediterranean. There are a few data points of larval bluefin tuna that have been sampled in the Mediterranean, and they plot around here. But as those fish have grown, there's really no evidence for extensive foraging in the eastern North Atlantic. There's very strong evidence that these fish have been foraging predominantly in the US side, in the western side of the North Atlantic, before maturing and coming here, and then presumably, potentially coming back down into the Mediterranean to spawn. But really strong evidence here that they overlap strongly with the western North Atlantic and not particularly with the eastern North Atlantic. Okay. So that's all things that you can do if you've got samples of isotopes from reference areas. You can build isoscapes or look at these comparative data sets. But what if you don't have any reference data? How can you use modeling to predict the isotopic composition? Or can you use modeling to predict the isotopic composition? Somebody here would say yes. So here's work that Sarah did while in my group as a PhD student, exploring how we can predict what the isotopic composition of primary production is likely to be if we know something about the biochemistry, or the isotope dynamics of, in this case, photosynthetic fractionation, or fractionation of carbon isotopes during photosynthesis. And the broad isotope systematics of associated with photosynthetic fractionation are quite well known. And effectively, we can predict the degree of fractionation, enzymatic fractionation, if we know a few terms, particularly the composition of CO2, the composition of carbon isotopes in dissolved inorganic carbonate, if we know something about the concentration of dissolved inorganic carbonate, and we know something about the cell size and growth rate. And these are terms that we can pull out of global biogeochemical models. So if we can pull these terms out of global biogeochemical models, then we can make some kinds of predictions about isotopic compositions in the global ocean. So here's Sarah's model carbon isoscape that estimates the isotopic composition we expect for annual average biomass-weighted primary production in a global context. And certainly we can also combine that with estimates of terrestrial carbon isotopes, and here those two things are combined on the same color scale. And one of the nice things about this is where you can see the land, then you would expect a big land to sea gradient in carbon isotope compositions. Where you can't see the land, then the average primary production on land is a relatively similar isotopic value to the average production in the water. So there you can see straight away where you'd expect a big land-sea gradient in carbon isotopes and where you wouldn't. Okay, but what can we use these for? So I want to show you some data sets we pulled for sharks. And we were interested in foraging patterns of different kinds of sharks globally. So here we're comparing oceanic sharks with coastal reef sharks, or coastal sharks. And the question we were particularly interested in is, can we tell whether sharks largely forage in the area where they were caught, or are they foraging large distances? How do we explore that? Well, this is another version of the model carbon isoscape, but now instead of looking at a continuous scale, we've simply combined the isotopic compositions for different biogeographic provinces. So as the isotope values of carbon, we expect in different biogeographic provinces, and the circles here are sharks that we have sampled. We have about 5,000 sharks sampled in different regions. And very simply, what Chris Bird, who was another PhD student at the time, did was to look at the difference between the carbon isotope composition of the shark and the area where it was caught, and to look at that for different large traits of sharks. So on this plot, you've got the distance from the equator on the x-axis and the isotopic composition of carbon on the y. These black values are the isotopic compositions of plankton that we predict from Sarah's model in the regions where the sharks were caught. And these grey values, which again haven't rendered perfectly, are the shark values. And so you can see for shelf sharks, we see a really clear relationship between the latitudinal gradient we predict for the plankton and the latitudinal gradient that we actually recover in the sharks. And that largely implies that the sharks are foraging in their local region. But for oceanic sharks, we see something very different. For oceanic sharks, the sharks that are caught in relatively tropical waters have much lower delta-13C values than you would expect. And that they have delta-13C values that are consistent with foraging in a latitudinal band around about 30 degrees, 30 to 40 degrees. So this this makes sense. It is the areas of highest production. It's where most of the global longline fisheries are. And it's effectively telling you that oceanic sharks found in tropical waters are not there to feed. But that has strong implications for construction of marine protected areas. If you construct marine protected areas with the intention of providing protection to oceanic sharks, and you construct those in tropical waters, you are not conserving the habitat of those sharks. The sharks are largely foraging in the areas where the fisheries are. And of course, it's a lot easier to establish an MPA in an area where there isn't a large industrial fishery. But that's not going to protect oceanic sharks in that area because they're foraging where the fishery is. Okay. One of the nice things about having a model is that as well as exploring space, we can explore time as well. So here we see, again, the model carbon ice escape, but now over months as well. So although I've rendered it quite coarsely to make it not too big, but you can just about see how primary production blooms propagate through time and create big changes and big pulses in isotopic compositions. That's jumping a little bit too much to see that. But the point is we have time and space. And having a prediction of isotopic variation in time and space allows us to really delve down into the mechanisms explaining isotopic compositions that we measure in animal tissues. So here's some work we finally managed to get published. This is a blue whale skeleton that is now hanging in the central hall of the Natural History Museum in London. Here is the whale when she died in 1891. She live stranded off of Ireland in 1891. So one of the nice things, of course, about a blue whale and other baleen whales is that we have baleen plates. So these are effectively hair. And that baleen grows continuously. And it grows relatively fast. And it's protein. So we can measure the isotopic compositions across a baleen plate, and that's going to tell us something about what that animal was doing while it was growing its baleen. So here's a baleen plate from a blue whale. The one that we've analyzed is around about that long. And we sample it at 1 centimeter resolution from the growing tip, so that's the most recent, to the tip that's wearing away, that's the oldest. And we end up with a track or an isotopic record of carbon that looks something like this. So I can see that I can almost see, if I squint, that there's variation in carbon isotopes. But what does it mean? How do I actually work out what is responsible for that variation in carbon isotopes? That could be movement, or it could be temporal change. So what I'm going to do is I'm going to take my temporal and spatial carbon isotope model and put a model whale on top of it. So I can write an agent-based model. I can almost write an agent-based model in a very ugly way. I can write an agent-based model to make my whale move, or forage and move across, in this case, the North Atlantic. So this is effectively what I'm doing. So here I've got some relatively simple rules that make my whale move and conduct different kinds of behaviors. And this is now the isotopic composition that I expect that whale to be ingesting as it's doing different kinds of behaviors in different places and different times. So effectively, we're now looking at a baleen plate growing and recording a particular isotopic composition at, in this case, daily resolutions, as it's doing different kinds of movement pathways. And because this is a probabilistic model, we can run this tens of thousands of times with lots of different movement trajectories, and we'll produce a suite of different predicted isotopic compositions that reflect different movement patterns. And so this is outcome of doing that. So red is, this hasn't rendered terribly well, but the red is the measured isotopic profile, and the grays are a suite of simulations, some of the best fitting 10% of 1,000 or so simulations that we ran. So we can take those best fitting simulations and explore what they mean. And in this case, for this whale, we've got a period of effectively residency in relatively warm waters. So this whale is staying in the same place for at least a year, and then conducting some seasonal migrations. And I'm going to take the seasonal migration part, and we can explore what is the likely movement pathway during that seasonal migration. And so effectively, we're now bringing that whale back to life. And these are the most likely monthly locations during 5 years of seasonal movement. And I think this is nice, that we can actually do quite a lot of reconstruction of movement just from stable isotopes, just from measuring along a baleen plate, if we know quite a lot about how isotopes work in time and space. Okay. Right, I'm going to go as fast as I can through the next bits. So one thing that we've been working at quite a lot at the moment is how we can use this spatial information locked up in isotopes to say something about traceability and food systems. And isotopes are used a lot in food forensics in terrestrial world, but not so much in marine systems. We can get into that later, why. But the concept is exactly what we've been talking about. If we measure the isotopic variation of fish in known regions or known fisheries regions, then we can build a database, a reference database, that allows us to probabilistically assign where any bit of fish that's been caught and traded is likely to have come from. And that means we can test whether eco-labels that says this fish was caught sustainably from a fishery in the Barents Sea, is that actually true? Or is it more likely that this has come from somewhere else? So just a brief, this is what we've been doing in undergraduate practicals at the moment. So this is now fish and chips, cod. The colours here are cod that we've caught from fisheries, from research fisheries in known areas. And the black dots here are fish and chips that we've sampled in various fish and chip shops around the UK. And the overwhelming majority of cod sold in fish and chip shops is coming from either the Barents Sea or the Norwegian Sea, which is what we'd expect, because 95% of all cod sold in the UK comes from the Barents Sea. But we can identify a few points that are likely to have come from, in this case, probably the Celtic Sea, probably Iceland. But we can, as long as we establish our reference populations, then we can do this for almost anything. And if we've got an isoscape, then we can go one stage further, and this is now doing continuous assignment, in this case for scampi. Most of the scampi, or nephrops, or langoustine, or whatever you want to call them, sold in Europe are actually caught around the UK waters. So we can use our UK isoscapes. And now each of these is now an individual scampi bought in a shop or bought in a supermarket, and the green areas are the most probable areas that individual prawn, that individual langoustine came from. We can sum those areas up and say what's the likely areas that are contributing to supermarket sold scampi at the moment. I used scampi in this case because it was easy, again, in an undergraduate practical, but you could apply this to any fish product or seafood product that you want, if you have an isoscape to compare it to. Okay, final thing. I apologize, I've gone on too long, but this is what I'm doing at the moment, mostly, which is trying to use isotopes to explore ecophysiology and fish responses to climate change. Now, we know that we've stored up all kinds of problems for the immediate and long-term future in terms of the ocean storing heat and continued warming of most ocean systems. There are various predictions of how fish and fisheries are going to respond to that warming. And those predictions are largely based on the metabolic response of fishes to increased thermal energy. And there's quite a lot of controversy as to how effective those models are. But they're all based on basically the metabolism, the metabolic response of fish to temperature change. And broadly, we can say, okay, basal metabolism will increase as a function of temperature up to some point at which systems start to become limiting. And we can try and explore what the metabolic response to temperature is likely to be if we know something about the thermal performance of different metabolic measurements. But at the moment, pretty much all of our understanding of the metabolic response of animals, fishes to temperature is based on measuring the standard metabolic rate, or the lowest metabolic rate we can measure for a fish, and the maximum metabolic rate, effectively how fast will the fish move when you poke it with a stick. And we can measure the difference between those two measurements as a function of temperature, and the difference between that is for some people seen as aerobic scope, and in some models aerobic scope is seen as a prediction of how fish are going to respond to temperature. But this is a very controversial model, and it also assumes that what you measure in the laboratory is actually what fish do in the field. And of course, in the field, fish have a whole range of other responses, a whole range of other things, energy-demanding things that they're going to do. So what we actually want to know is, what is the metabolic response, the energetic response of fish in the wild to changes in temperature? So we really want to know what the field metabolic rate of animals is. And I'm going to race through this because I have to. So what we've been starting to look at are otoliths. Otoliths are calcium carbonate structures in the fish ear. And as we all know, otoliths are used for aging fish in fisheries and enormously important structures. But it also means that we have a large amount of otoliths in archives. And that's important later. But their otoliths are calcium carbonate, which means that they're made of carbon. And the fish sources that carbon from two possible places. There's seawater or environmental carbon, and there's respiratory or diet carbon. And these two things are very distinct isotopically. They mix in the blood of the fish, and that mixture of carbon in the blood of the fish is what is sourced to go into the otolith. As you increase your metabolic rate, effectively what you're doing is you're increasing the rate at which you respire your diet, which means that you increase the proportion, at least theoretically, you increase the proportion of respiratory carbon in the blood. And we should be able to measure that by a mixing model that allows us to basically solve how much of this there is in the otolith. So we've done an experiment. Here's temperature of little juvenile cod in a tank, temperature, and this is the proportion of metabolic carbon in the otolith. And you can see that there's a very clear relationship between the proportion of metabolic carbon recovered in the otolith and temperature. And of course, temperature is increasing the the oxygen demand. And actually, we also measured the oxygen consumption rate of these fish. So we have a calibration, at least for a large portion of the likely oxygen consumption range. We have a calibration between the proportion of respiratory carbon in the otolith and oxygen consumption rates. And that means that this value, the proportion of respiratory carbon in the otolith, is a proxy for the absolute field metabolic rate. And it's something we can measure back in time. The thing that's really beautiful about otoliths is when we measure the carbon isotope in the otolith, we also get the oxygen isotope. The oxygen isotope in the otolith gives us a measure of the temperature that that individual fish was experiencing at the same time as it was growing the otolith. We also have the growth rate of the fish from otolith increment analyses. So for any individual otolith, we have a measure of the temperature that that fish experienced, the field metabolic rate it experienced in that temperature, and how much it grew. That's an incredible resource for understanding bioenergetics and temperature. I'm just going to show you a few examples of how we can explore this. We've started to explore the relative field metabolic rate of a large range of taxa to start looking at the sort of macroecology of field metabolic rate. And here we've just ordered field metabolic rate as measured from carbon isotopes and otoliths across nearly 100 taxa. And this is now colored by depth range. So unsurprisingly, deep water fishes have much lower metabolic rates than pelagic fishes. But we can start to explore this with a lot more trait-based analyses of, are there certain traits of fishes that mean that they have different field metabolic responses. I want to give you a couple of slightly more exciting examples of how sensitive this can be. So here we asked, do different morphotypes of fish inhabiting the same waters, do they experience different metabolic sensitivities to temperature? So here we have cod, juvenile cod that are caught in fjords in Norway. And we have two morphotypes, two genetically distinct morphotypes of juvenile cod. There's a fjord-type cod, which basically always stay in the fjords. And then there's North Sea cod that get advected in and live in the fjords as juveniles and then advect out again. So here we're measuring, from the otoliths alone, we've got the temperature that the individual cod experienced and the relative metabolic rate from the otoliths for two different populations. The fjordic cod are generally colder water adapted because the fjords are relatively cold compared to the North Sea. North Sea cod are warm adapted. And what you can see is these two genetically distinct populations of the same species living in the same water have different thermal sensitivities of field metabolic rate. So this has really interesting implications for predicting the responses of populations to temperature change. And the North Sea cod are more sensitive to temperature. They have a higher slope of metabolic response to temperature. But they also choose, the distribution of temperatures that these individuals have chosen, they choose to live in the warmest waters possible. Whereas the fjordic cod have a less sensitive to temperature and choose to occupy colder waters. As these waters warm, the metabolic advantage of the fjordic cod in the cold waters is going to be lost. So we end up with a prediction of how these different morphotypes are likely to respond to warming. We're measuring about 2 weeks of temperature and metabolic rate in this otolith milling. An MPA example. So here is in South Africa, Tsitsikamma National Park has been a no-take zone since 1964. So it's a really well-established marine protected area. And this is a fish called the red Roman. So we've been looking at the metabolic rate and temperature experience of red Roman in Tsitsikamma National Park here and in a fished area, Port Elizabeth, a little way over here. So this is now the same types of plots, temperature and metabolic rate in Port Elizabeth, the fished area, and Tsitsikamma National Park, which is the MPA. Now the actual metabolic response to temperature is the same in these two regions. But what you can see is that in the fished area, there's a really small temperature range that the fishes are actually choosing to experience. And it's the same temperature as basically the highest metabolic rate that these fish experience. In Tsitsikamma National Park, we see fish that are in water that's colder than their optimal and warmer than their optimal. They're experiencing a much wider range of temperatures. Why is that? Well, in the national park, there's a hell of a lot more of them. They're not fished. So they're forced, I should say these are very, very, very small movement animals. They're very territorial and they don't move more than a few meters. So in the MPA, there is a paucity of areas that are the perfectly optimal temperature. In the fished area, there's not as many fish and they can choose exactly the optimal temperature. So we're starting to see how the population density is influencing firstly their temperature choice and their metabolic response to that temperature choice. Back to tuna, because Why not? Tuna are cool. So again, bluefin tuna. The otoliths of bluefin tuna are used to determine whether juveniles came from the Mediterranean or the Gulf of Mexico, largely because Mediterranean waters are cooler than the Gulf of Mexico, and that leaves an expression in the oxygen isotope composition of the otolith. But we can also look at the carbon isotope composition of the otoliths that have been measured in these bluefin tuna and see what the relative field metabolic rate is for these two populations of tuna as a function of their experienced temperature. So this is Mediterranean origin tuna, and this is Gulf of Mexico origin tuna. And effectively, you can see that there's a parabola. So the highest field metabolic rates are experienced by fish that are in water temperatures of around about 28 degrees. That happens to be pretty much the warmest temperature that bluefin tuna from the Mediterranean ever experience, and it's close to the coolest temperatures that tuna from the Gulf of Mexico experience. What does that imply as waters warm? Well, it implies that in the Gulf of Mexico, a large proportion of bluefin tuna are in waters that are already warmer than the temperatures that are going to be conducive to maximum growth. So already, bluefin tuna from the Gulf of Mexico are smaller and older when they exit the Gulf of Mexico as juveniles than they are when they exit the Mediterranean. So I think this is already part of that metabolic temperature optimal that we're seeing at around about 28 degrees. As the Gulf of Mexico warms, we expect bluefin tuna to be older and smaller and probably have greater mortality because it's taken them longer to grow. And then, like the last one, which is plaice in the North Sea. Plaice in the North Sea have a really interesting migration. So these are they migrate to spawn in winter and feed in the central North Sea in summer. And you can see that clearly from the condition. This is condition by month for males and females. So at this point, around about May, you see females are growing and growing and growing, maximum condition, and then they start to spawn, lose their condition. So what happens to the metabolism of these fishes as they do that? So Joe Jones, a PhD student in my group at the moment, he's measured about 400 individual plaice, and we've measured them every month. This is using otolith archives. We've measured the field metabolic rate and experience temperature as a function of month for these plaice. And what you can absolutely not see is a clear relationship between temperature. So for these adult plaice, temperature is not a strong driver of field metabolic rate. Most of the predictions use temperature as the main driver, main prediction of field metabolic rate. What we're actually seeing here is a period of high metabolic rate in cold waters. Paradoxically, for plaice, cold waters are in summer because they go into the central North Sea where it's colder. So we see high metabolism in summer when they're cold and high metabolism in winter when they're warm and spawning. And this is effectively, this is elevated metabolism for spawning, elevated metabolism associated with feeding. For a lot of fish, feeding the highest metabolic rates that fish experience in the wild is often associated with eating a lot. This is a high metabolic cost of digestion. So I think what we're seeing here is fish feeding, trying to put on as much condition as they can and elevating their metabolic rates in cold waters and then resting and then expending that energy during spawning. And we've got enough sensitivity in these analyses to pick that out. But what's really nice, of course, with an otolith is we can measure the outside part of the otolith and get the most recent metabolic rate as the adult, but we can go back in the otolith and measure the metabolic rate of the same individual as a juvenile. So here's a subset of those data. This is year 1 of those individuals, and this is year 4 of the same individuals. So in year 1, there's a clear relationship between temperature and metabolic rate. So high temperature leads to high metabolic rates when all they're trying to do is grow because they're year 1 fish. When they're adults, that predictive power of temperature to predict metabolic rate is gone because they're doing a whole lot of other behavior and temperature is not predicting metabolism. So this has really strong implications for how you might use temperature to predict not only the behavior, production, distribution of fishes in the future. There's a lot of interesting physiology and biology explaining how fishes are using energy at different stages in their life, which can be extracted from the isotopes of incrementally grown tissues. Oh, didn't need to say that. Okay, I have finally finished. So what I want to come back to here is just what an enormous range of information you can extract from this crazy, simple difference of one extra neutron in a carbon isotope. We've done a huge number of different things that you would think are impossible to measure or impossible to infer just because there's one extra neutron in this atom. And I do this a lot, and I still come back and think that's just beautiful and amazing. So it's a wonderful tracer, and it's one of these things where your imagination can really take you in different ways, and you can learn some surprising things because it's a relatively open field. And we just opened the book on this field using isotopes of field metabolic rate. There's an insane number of things we can do with that. So we can use isotopes to explore trophic ecology, spatial ecology, movement, connectivity, historic ecology, and ecophysiology. To do all of those things, we do also need references, and particularly isoscapes. And building isoscapes also allows you to look at biogeochemical mechanisms, pollution tracking, opens you up to doing forensics and traceability, just an enormous range of different questions that you can explore. And whatever animal, plant you decide tickles your fancy, you get to work with if you understand isotopes. And I will finally finish. Thanks very much.
Does any of you have questions?
Clive, thank you. That was great. Really enjoyed it. I'm fascinated with the idea of what you're doing at Tsuchikama, because what you're doing is implying that there's habitat selection that's entirely density dependent, which is sort of something that's been thought about a lot in the context of MPAs for a while, but there's not much data around. My question is, those fish that were in suboptimal areas, were they smaller ones?
I don't know that, but we could work that out. We have the growth data and the size data for all those individuals.
Yeah, because given the territoriality, that's what you'd expect to see, isn't it?
Yeah.
Which is serious implications for what people make predictions about in terms of spillover.
Yeah.
One other one that I wanted to ask in general is, if otoliths are so great, why bother with eye lenses?
Because eye lenses are organic proteins and otoliths are minerals. So it's very hard to extract the dietary data back in time from otoliths. There's so little organic component in the otolith that it's really hard to do that. The other thing is the eye lens. You can use eye lenses on sharks, which don't have otoliths, or squid. So you can work on some things that you don't have any other options for going back in time.
I have a question about the otoliths. Can you do a resolution year by year or seasonal resolution?
If you have the money, and depending on the species, you could do, like on a juvenile cod, you could do 2 weekly resolution if you really wanted to.
Because an otolith is quite small, but do you have enough quantity?
Oh, we need 20 micrograms of powder for an analysis. Okay. So you're limited by your patience and your budget.
Okay.
But yes, there's a hell of a lot of tissue in an otolith in comparison to how much you need to measure. So we've been doing it with conventional gas source mass spectrometry. You can also use secondary ion mass spectrometry, which is basically a beam-based analysis. And at that point, you can get a measurement from a 20 micron beam size, which means that you can get really, really crazy temporal resolution if you absolutely wanted to. At that stage, it's going to get really difficult to interpret, but you could. Yeah.
other questions?
Thank you, Clive, for your super interesting seminar. I had a question already when I was reading your paper about the use of jellyfish as, let's say, tracers to desume the isoscapes. How do you account for the different lipid content of the jellyfish if you use this large number of animals, and so they may be in different reproductive stages and therefore have different lipid contents? How do you deal with the lipid content in general? And how do you deal with the differences? I have seen that you have used different jellyfish groups, and this means, for example, if I just think of the difference between semiostomes and rhizostomes, which have different, probably different feeding regimes, feeding patterns, how do you deal with all this? Thank you.
No, of course. Really important questions, absolutely, and potentially hard things to cope with. So we only use bell tissue for starters, and in the statistical framework that we chose to build the isoscapes is chosen specifically because of its power at identifying and separating spatial from non-spatial components of variance. So we can, so obviously we track the lipid content from the carbon-nitrogen ratio in the muscle. So we can in the bell tissue. So we can use the CN ratio if we want as a variance component that we want to extract from the database. In fact, there was relatively little variance in the CN ratio of the bell tissue that we sampled, and we were sampling specifically to try and avoid problems associated with allocations of lipids to gonads or whatever. So we did an okay job of that. However, no, we put species as is one of the random terms, the non-spatial terms. So there may well be some physiological components about slightly different protein compositions or protein lipid compositions in the bell tissue that would be contained, if they're species level, they would be contained in the species effect. So for instance, the barrel jellyfish comes out as radically different in carbon, and I'm sure that quite a lot of that is associated with differences in the protein content of the bell tissue. But that will be taken out and ascribed to the species level effect. So we've taken that out of the spatial component. And that's the advantage of using the inlet approach, that you can identify things that you expect to have to be a source of non-spatial variance and remove those from the spatial terms. It's basically the equivalent of a random effect term in a mixed model. But that's how we deal with that.
No more questions?
Talked for too long.
Well, thank you very much for
Thank
the
you very
seminar.
much. Thank you.